Hello everyone. In this class, we learn how to enumerate RBCs. Aim of this experiment will be to determine the total RBC count by manual method. And principle of this experiment is the number of RBCs in a known volume of diluted blood is counted and the number of cells in undiluted blood is calculated and reported as the number of red cells per cubic millimeter of whole blood. The apparatus required are microscope, Newbus counting chamber, RBC pipette, cover slip, and RBC diluting pipette, which is called as Hames fluid. Components of Hames fluid and their functions. It contains sodium chloride 0.5 grams, sodium sulfate 2.5 grams, mercury chloride 0.25 grams, and distilled water 100 ml. Sodium chloride, it helps in maintenance of osmolarity, whereas sodium sulfate, it prevents aggregation of the RBC, whereas mercury chloride, it acts as a preservative, that it acts as a antifungal and antibacterial and hence prevents growth of microorganism, whereas distilled water, it acts as a solvent. The procedure is clean the pipette, cover slip and Newbus chamber thoroughly, then place the cover slip on the central platform of the Newbus chamber, then take adequate RBC diluting fluid in a watch glass and under aseptic precaution prick the finger, that is middle or ring finger, and suck the blood up to the 0.5 mark. Then suck the RBC diluting fluid up to 1 and 1 mark. Then hold the pipette horizontally and close both the ends of pipette, then gently mix the contents of bulb by rolling between the palms. Then discard the first 2 drops of fluid from the pipette. Then charging is done. Charge the Newbus chamber by placing the tip of pipette on the surface of the chamber, touching the edge of the cover slip at an angle of 45 degree. Allow the diluted blood to flow under the cover slip by capillary action. Take care to avoid entry of air bubbles and flow of fluid into the gutters. And recharge if the air bubbles are present or if there is an overflow, which is called as overcharging. And allow the cells to settle down for 2 to 3 minutes, then place the chamber on the stage of microscope. This is how under the low power the Newbus grid will look like. Okay, so with the low power you'll be able to appreciate all the RBC counting chamber in the central square, that is 5 intermediate or middle squares. Observe first under the low power and ensure equal distribution of RBCs. Focus under high power and start counting the RBCs in 5 medium-sized RBC squares. To avoid reduplication in the counting process, apply L rule. The number of RBCs in each of the 5 square should be approximately equal. That is, the difference between the maximum and minimum should not exceed 20. This is how under the high power you'll be able to see one of the intermediate square in which you'll be able to see all the 16 squares. And so these are the RBCs, how they look under microscope under 40x. Okay. Then you have to count the number of RBCs in each of these chamber in a zigzag manner like this. So, and to avoid overcounting, you have to first count the number of cells which are there in the box and which are not touching the middle line. Okay. So if the cells are touching the middle line, the cells which are touching the L of the corner should be considered for this particular box only, and the cells which are touching the opposite of the L, it should be excluded from this box, and this is called as L rule, and this is done to avoid overcounting of the RBCs. So dilution obtained with mixing of the fluid is 1 in 200, because the volume of the bulb is 100, because it is not 101, because the 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 diluting fluid which is present in the stem, it will not take part in dilution. So 100 volumes of diluted blood contain zero point 0.5 ml of whole blood. So 200 volumes of diluted blood contains 1 volume of whole blood. Therefore, dilution here is 1 in 200. So then we have to calculate the number of RBCs in 1 cubic millimeter of diluted or rather undiluted blood. So for that, the area of one medium size square should be calculated. That will be 1 by 5 into 1 by 5 is equal to 1 divided by 25 square millimeter. So thus we will be counting such RBCs in 5 such medium size squares. So 1 by 25 into 5 will be 1 by 5 square meter. So then volume of such 5 medium size square will be 1 by 5 into 1 by 10 is equal to 1 by 50 cubic millimeter. So 1 by 50 cubic millimeter of diluted blood contains n number of cells. That is the number of cells we have counted in all the 5 intermediate squares. So therefore 1 cubic millimeter of diluted blood contains 50 into n cells, and since we have diluted the RBCs by 200 times, so 1 cubic millimeter of undiluted blood contains 50 into 200 into n. Therefore, RBC count is equal to 10,000 into n. If we have 100 cells in each of the intermediate square, so then the RBC count will be somewhere around 5 million cells per cubic millimeter. The normal RBC count in males is 4.5 to 5.5 millions per cubic mm of blood, and in female it ranges between 4 to 4.5 millions per cubic millimeter of blood. And in the end, the report has to be written like this: name, age, gender, occupation, and the RBC count, for example 5 million cells per cubic millimeter of blood. Some of the questions which are asked usually in the Viva are: what is the function of bead in the bulb? The bead acts as a stirrer, and free rotation of the bead in the blood signifies the pipette is dry, and bead are distinguishing feature, that is red bead for RBC pipette and white bead for WBC pipette. Why the fluid in the stem is discarded? The fluid from the stem portion of the pipette is discarded because the actual mixing is taking place only in the bulb, and stem contains only the diluting fluid. Since the mature red cells do not contain nuclei, are they dead cells? The RBCs, though without nuclei, are not dead in the usual sense. Since there are no mitochondria and other organelles, energy is not available from oxidative phosphorylation, that is through Krebs cycle. However, they contain a number of enzymes which control important functions. The cells contain all enzymes necessary for energy supply from anaerobic glucose metabolism via Embedon-Meyerhof pathway, that is hexose monophosphate shunt. Glucose enters the RBC by an active process which is insulin dependent. What are the other uses of Newber's chamber? It helps in finding out RBC count. Apart from RBC, it is used for WBC count, platelet count, and also cells in cerebrospinal fluid, peritoneal, pleural fluids, etc. What are the physiological and pathological variation in the RBC count? Increased RBC count is observed physiologically in males at high altitude, at high temperature, low oxygen tension, that is hypoxia, after exercise, and in infants. Pathologically, it is seen in polycythemia vera, hyperthyroidism, and Cushing's disease or syndrome in case of dehydration, chronic lung disease, or congenital cyanotic heart disease. And decreased RBC count is seen in all the types of anemia. So this is in brief about enumeration of RBC. Thank you.